IDIOTYPEs, and they are being often used for the antibodies. So what are these terms means? Let's talk about that today in this particular video. So before starting and talking about this isotypes, I would like to give you how the antibody is being produced inside our body. We know that whenever a pathogen or an antigens it's tried to invade inside our body, there is a certain kind of a immune response is generated by means of certain lymphocytes, let's suppose the B cells. So B cells, whenever they found in the circulation that there is some kind of a foreign pathogen or a foreign substance is circulating, they will bind to that pathogen with the help of their B cell receptors, right? So this is the B cell receptor which will bind to these antigens molecules, right? Or you can say specifically the epitope of these antigens, they will bind to it, and this will then trigger a signal. This will give a signal to the B cells for them to activate, and there are certain kind of a genes or the class switching of these genes take place inside it at the molecular level. So here I'm not talking about that. That is the separate topic, and that's a huge topic, so I'm going to cover up in the separate video only, right? That is about the class switching and recombination and the VDJ recombinations, right? So that we are not talking about it. So that kind of a molecular signal will be sent inside, and then in turn there will be a one more signal will be generated from the T helper cells. The T helper cells, they are also circulating and in the blood, and whenever they found that there is a binding of the antigen and the B cell receptor happens, they will get activated and they will start secreting certain kind of chemicals known as cytokines. These cytokines in turn will give a signal to the B cells that yes, now you should start proliferating and should start dividing, and they will start forming lots and lots of plasma cells and lots of memory cells. So what these plasma cells do, the plasma cells start secreting lots of antibodies. We know that plasma cells are basically the factories of antibody. They will start producing lots and lots of antibodies, and these antibodies will then try to neutralize all these antigens which they found in the circulation, right? So they will neutralize them, or they will generate certain kind of immune response so that other immune cells can come and will kill these pathogens, right? What the memory cells do, they keep the memory of these pathogens or the antigens by keeping the antibodies on their surface which are being produced specific to these pathogens, right? So whenever in future these same pathogens going to infect our body, the memory cells already have antibodies against them, and they will trigger a rapid clearance of these pathogens, right? So that is how the whole concept, how the immune system works, right? So now coming back to the antibodies, since we are talking about isotype, idiotypes and the allotypes, so let's talk about the antibodies. So we know that antibody structure is made up of 4 polypeptide chains, right? 4 polypeptide chain is present, out of which 2 of them is the heavy chain and 2 of them is the light chain, right? So if I draw it here, so these are the 2 heavy chains which are present, and these are the 2 light chains which are present, right? And you must be knowing that in these heavy and the light chains there are 2 specific portions are present. The uppermost portion of both these heavy and the light chains are known as a variable region, and the lowermost regions, this one is known as the constant region, right? So why I'm talking about these constant and the variable region here, because mainly these terms which we are going to talk about it, they are named because of the slight difference in the amino acid sequence of these variable region and the constant region only, right? So that's why these terms has been given to them. So it is very essential you should know that what are the variable and the constant region in them, right? So let's now begin it with the isotypes, right? So if this is the antibody structure, as I've told you that this is the heavy chain, right? This whole thing is the heavy chain, the 2 heavy chains, and these are the 2 light chains which are being present, right? So sorry, this is the heavy chain, this is the light chain. Yellow one is light chain, yellow and green are light chain, and red and blue is the heavy chains. right? And if you talk about this portion of the this is the variable portion of the heavy and the light chain, and this is the light portion, light chain constant region, and this is the constant region of the heavy chain, right? On either side the same way. So depending on which type of heavy chain is present, there are 5 different classes of antibodies are present in our body. So if we talk about humans, we humans have 5 classes of antibodies in our body which are keep on producing. Now why these 5 different classes of antibodies are being produced, or why there are different 5 classes are being there? Because there are different 5 classes of heavy chains are being formed. 5 different heavy chains are being produced in our body, right? So depending on which type of heavy chain is present in the antibody, there are different 5 classes of antibodies are being produced. For example, we have 5 different antibodies which are IgG, IgA, IgM, IgD and IgE, right? So IgG will have a with heavy chain, gamma heavy chain. IgA will have a alpha heavy chain, M will have a mu heavy chain, IgD will have a delta heavy chain, then IgE will have a epsilon heavy chains. So these heavy chains in the structure of these antibodies are different in the different classes of the antibodies, and that's why they are being named accordingly. Gamma ka G, alpha is A alpha, and mu M, delta D, epsilon E, right? Accordingly they have been given the name. Now all these 5 different classes, they have a different structure because of the difference in the heavy chains which are present in them, right? So if I talk about the structure, you can see here that these are the different structures form in which these different antibodies are being present in our body. For example, this is a monomeric form which is mainly being mainly the IgE, IgG and IgD. They are mainly present in our body in the monomeric form, while the IgA, they are present in the dimeric form. So 2 monomers are being attached by means of a J link, and that's how they are being present in the dimeric form. They are being mostly found in our body, right? And this IgM is mainly present in the pentameric form, right? And also they all have a different functions, and in different amount they are present in our body, right? So these different isotypes, so the classes is basically the isotype. So you can also say that not only 5 classes but 5 isotypes are present. So any word you can use, isotype or a classes. So isotype means classes only. So you can say there are 5 isotypes of antibodies which are present in our humans, right? And all these 5 isotypes have a different role, and in different amount they are present. For example, IgG is abundantly found antibodies in our body, right? And IgD is very less present, like 1% is only present in our body. All right. Also their functions are different. For example, Ig is mainly responsible for the allergic reactions, while IgA mainly is you will find in the secretion, in the secretory forms, like the secretions of breast milk, right? So you will find IgA. So all of them will have a different functions, but only the difference, although the structure is same, they all have the same 2 light chains, 2 heavy chains. The light chains will be almost same in all of them. The only difference is in the heavy chain, right? So all of them will have a different heavy chains, and that's how accordingly their isotypes has been named. So these are the isotypes, right? Now let's talk about the allotypes. Now allotypes, allo means word is comes from the alleles. Alleles, we know that alleles are the different versions of the genes. For example, if we talk about the hair color, right? So different individuals have a different hair color because we have a different versions of that hair color gene is present in the different human being, right? So some will have black hair, some will have a blonde hair, some will have a brown hair, right? So everybody hair color is different, although the hair gene is present is same in all the human species, but individually to individually, why the hair color is different? Because the allelic version of that hair color gene is different in different individual. For example, here if we look about this is the individual 1, let's suppose, right? So this is individual 1 and this is individual 2. Individual means a person 1 or a human being 1, human 2, let's suppose, right? And both of them have a same class of antibody, IgG1, IgG1, and this kappa stands for light chain, right? So if you let me give you an additional information here, that heavy chains, as I have told you, that heavy chains which are present in humans, there are 5 classes of heavy chains are present: alpha, delta, mu, epsilon and gamma, right? So these are the 5 different classes of heavy chains are present. Similarly, the light chains, we have 2 different light chains are present. One is kappa and one is lambda. Mostly the kappa is found, lambda is found very rarely, but these 2 classes of the light chains are present in us, right? So here in this example, if we have taken the 2 individuals and both the individuals have the IgG1 class of antibody, if we look at the IgG1 class of the antibodies, so we are considering that their light chain is the same. They both are the kappa light chains are present in them, right? So what is the difference in them? We are saying the isotypes, the subclass is also same, IgG1 is there, and the light chain is also same. So what is the difference? So just have a look in these pictures of both of them. You will find that these, what are these which is shown here with the blue and yellow, and here yellow and red? What is this differences? These are basically denoting the amino acids, right? Different amino acids. Since polypeptide, since these are the heavy chain is what? This is a polypeptide chain. Polypeptide is a polymer of amino acids, right? So they are made up of amino acid sequence only. So if you see here that they are almost same, they are the same classes, same light chain is present, but in the heavy chain constant portion, this is a constant portion, right? Heavy chain constant portion. So if you look at here you will find there is a slight difference in the amino acid sequence of both of them. So this slight difference in the amino acid sequence will lead to a structural difference among both these antibodies, right? Although the class is same, but still there will be a difference in the structure because of the subtle changes in the amino acid sequence among both of these antibodies at their constant portion of the heavy chain region. So that these are called as the allotypes, right? I hope this is clear to you. So there is no such difference among them. Only difference is there is a slight difference in the amino acid sequence at the constant portion of the heavy chain region, right? So that's why they have been called as the allotype, because this will lead to structural changes in both these antibodies. Although the functional difference will be very slight, difference will be there, the main difference is because of the structures, right? So that's why they have been named as the allotypes here. Now let's talk about the idiotypes. Now, so idiotypes, let's look at with this particular diagram. So if this is an antibody structure and this is a pathogen which is trying to invade inside our body, and this is a specific antibody against this, right? So if I zoom this and let you know what is the how the antigen binding portion which is over here and this PAP portion of this antibody, how it looks like, right? So if this is the antigen and this is a epitope, I hope you might be knowing that the entire antigen never binds with the antibody, or you can say antibody never binds with the entire antigen, but it binds to a specific portion on the antigen which are known as the epitopes, right? So this is a one particular epitope with which this antibody will bind, right? Have a specific antigen binding site present on it, right? So this specific antigen binding site which is present on the antibody, this is known as what? Paratope, right? So, and here if you can see these red ones, white one, what are these? They will also interact with these antigen when it is going to bind. Just imagine it, when this is going to bind here, which this is going to fit here, these portion will also interact with this antigen binding portion of the antibody, right? And these portions, what are these known as? They are known as idiotopes. What are those known as? Idiotopes, right? So this is also idiotopes. So they will also interact with these antigen portions, right? So along with these idiotopes and paratopes, what are these known as? Idiotypes, right? So this difference in the idiotypes will take place when it will take place, just think about it. Obviously it will take place when there will be a certain different pathogen is going to infect it. Let's suppose if this is antigen 1, and if let's suppose there is an antigen 2 which is now try to invade inside our body. Let's suppose its shape is somewhat, its epitope shape is somewhat like this, right? And against this particular antibody with which has a specificity, a specific antigen binding portion, that will going to interact, right? So if I draw it here, if this is that antigen and this is the epitope, okay, so this is the, let's suppose this, right, like this somehow. So this is how it will look like, right? So just imagine it, right? So if this is the light chain and this is the heavy chain, so this will be then the paratope, right, which is going to be bind to this epitope of an antigen, right? And here also there will be a different idiotopes will be present, right, which is also interacting with this antigen, right? So when this will bind, they will also interact. So together all of them are known as idiotypes, right? Although the antibody is same, let's suppose here also IgG is being produced against this particular pathogen, and here also the IgG is only produced against this particular pathogen, but because there is a specific different binding sites or different epitopes which are binding with different antigens, and there are different idiotopes are being expressed on these same class of antibody, this is known as the idiotypes, right? So they are known as idiotypes. Now the term will be used for these antibodies is that there are different idiotypes are present on the IgG antibody, right? I hope this is clear to you. So the idiotype is basically a difference in the amino acid sequence at the variable portion of the heavy and the light chain of an antibody, right? I hope this is clear, right? So in this example, as I've told you, like here you can see again that if this is mouse, let's suppose strain 1 mouse, okay, and this is also the same mouse, if we administer this antigen, inject this antigen 1 into this mouse 1, okay, and it will produce an IgG1 against it. So you can see that these are the different idiotopes which are present. The blue color it is mentioned here. So these are the different idiotopes against this particular antigens, right? Similarly, if we administer into the same mouse the antigen 2, let's suppose this is the antigen 2. Now the same antibody is going to generate. Let's suppose ID1 is only generating against this antigen 2 also. But here you can see there are different idiotopes are present on this particular antibody. Although the antibody is same IgG1, but the idiotopes, that is the antigen binding portion, or you can say the paratope and the idiotopes, they all are different in different antibodies against different pathogens, right? So they are known as, here the term we will use is idiotypes. So this is very simple, nothing much about it. In the isotypes, what was happening, if this is the antibody structure, in the isotype what was happening, the heavy chain is every time in all the different classes there will be a different heavy chain will be present, right? Depending on which class of heavy chain is present, the different types or the isotypes of antibodies present in our body, right? In case of allotypes, there will be a subtle change in the constant portion of the amino acid sequence of this heavy chain region, right? Although the antibodies will be same, so they will be known as allotypes. In the idiotypes, there will be a variable portion amino acid sequence changes is there, and that's why they are known as a idiotypes. So that is because of the different antigens which is going to being recognized by these antibodies, right? So we can summarize that all the heavy chain constant region which give rise to our classes and subclasses are expressed together in serums are called as isotypes. The variations in the heavy chain genes which leads to the functionally insignificant but structural differences, especially in the constant region of antibody because of the differences in the amino acid sequence, are called as allotypes, and the variation of the amino acid sequence within the variable domains on the heavy and the light chains are called as idiotypes. So this is all about the different terms, and thank you so much for watching. If you like my lesson, please do subscribe my channel MS Bio Academy, and please don't forget to share with all your friends. Let me know in the comment section down below if you have any queries. Bye for now. See you soon with the next lesson.